Now it is my pleasure to introduce our next speakers. They are from RheoSense. We have our research and development team with principal scientist Dr. Stacey Elliott, an application scientist Chrystian Ochoa, and our application scientist Sohelia Shabani, who will be presenting today on monitoring the structure of DNA with viscosity. Now, without further ado, it is my pleasure to turn it over to the RheoSense Research and Development team.
Okay, thank you, Eden. Can you just quickly confirm that you can see our presentation?
Yes, we can.
Okay, great, thank you. All right, so let me just move on here. Next. Okay. So as Eden said, we're going to talk about how we can use viscosity to monitor structural changes within DNA. And as you know, we talk a lot about in many of our other presentations about how viscosity is so sensitive to the structure. And like some of our previous speakers of today, we've spent a lot of time using viscosity to understand other types of biological molecules like the antibodies and proteins. And so we wanted to try out and see how can we use viscosity to understand structural changes that occur in DNA. So to sort of set the groundwork, I wanted to go over some basic information on the structure of DNA. And so I think everyone's probably seen this double helix picture that we have up in the corner here. And so generally this common structure that DNA will assume is coming from the base pair hydrogen bonding of the single strands. And this is the sort of origin of that double helix that you commonly see. Another thing to keep in mind about DNA is it's negatively charged or anionic. And so now you have some like charges along the backbone of the molecule that tend to influence the structure due to those electrostatic repulsions that you have. Commonly, DNA is composed of up to thousands of base pairs. So this means the DNA can have a very high molecular weight in the millions. And in fact, we did some studies with the the DNA that we're using, and we kind of estimate that the molecular weight of our molecules that we worked with is slightly over 3 million. And the nature of this in terms of how many base pairs you have is actually quite important when it comes to the different types of structural changes that we talk about. In fact, you have to have, in general, I think it's over 400 base pairs to see some of the transitions that we'll be monitoring. So to think about, the native structure of DNA, it's going to be this charged macromolecule, very similar to what you would think of when you think of a synthetic polyelectrolyte that has sort of an extended worm-like or coiled structure, extended due to the fairly stiff nature of this double helical structure, and then also from those electrostatic repulsions along the backbone. So how do you induce structural changes in DNA? There's various ways to do this. One of these, and this is sort of the first one that we thought we would explore, and I'll sort of explain how that didn't quite go to plan, but that's basically the introduction of small molecule drugs that can interact at various positions along that backbone of the DNA. Another is what was referred to as a condensing agent. These are often used to drop the viscosity by changing the structure of the DNA, and we'll go over what that means. And then there's basically just heating cooling cycles that will change the nature of the DNA. Not too dissimilar to denaturing of proteins, however, in this case, the thermal changes are actually reversible, although to actually achieve that return to the original state can be a little tricky, which we'll talk about as well. So just a quick discussion on the drug interactions, since things didn't quite go to plan for us. So certain drugs are designed to target intracellular DNA, which is why people study the interactions of various drugs with the DNA structure. And there are some different types of bonding that these small drug molecules that can have with the DNA backbone. These could be electrostatic, external or groove, or intercalative. So we sort of highlighted the intercalative type of interaction here because this is what we were hoping to study. And what happens with this type of interaction or association is that a molecule such as sumatriptan, so this is a small molecule drug, will insert itself in between the base pairs in the double helix. So why is that relevant? So when this small molecule inserts itself into the structure, theoretically what you would expect to happen, and this has been shown empirically as well, is that it would extend or elongate this double helical structure. So you're basically going to expand the random coil or worm-like structure that you have if you introduce enough of these small molecule drugs into the DNA structure. So you would then expect that you would see potentially an increase in solution viscosity as the molecule expands. And so here's sort of an image of, just to kind of clarify what I hinted at in a couple slides ago, and the overall structure that the DNA would assume. It's going to be sort of coiled like, where if you zoomed in, you would see each individual portion of the chain has this double helical structure, and then potentially if you're introducing a drug, it would insert itself there. The challenge is, in terms of trying to use viscosity with our technology to monitor any sort of structural changes due to the drug intercalation, is that, as I mentioned earlier, the DNA tend to be very high molecular weight. In our case, we estimated ours was over 3 million. Now, what that means is that because this is a larger molecule, larger than, say, the antibodies that people have talked about previously, where those are probably in the order of 150,000 or so Daltons, these million molecular weight plus molecules will shear thin even in the dilute limit. So that means that there's some significant deformation of that molecule, even in the dilute state. And this deformation coming from the shear can kind of obscure any sort of extension that comes from the intercalation of the drug. So it was very difficult for us at the rates that we were able to achieve to actually see this change. So we would need to somehow get to lower rates, or perhaps look at drug intercalation of a lower molecular weight DNA, so something below 1 million, maybe at 500,000. So we sort of moved on and thought, well, what else can we do with our DNA, and how can we use viscosity to monitor this? So I'd hinted at the condensing agents. So condensing agents basically do just that. They're introduced into the formulation to essentially collapse the DNA and cause it to form a more compact structure. And these compact structures, they can be composed of either one DNA molecule or multiple DNA molecules. And once you sort of shrink down the presence of the DNA and essentially drop the volume fraction occupied by the DNA, then we would expect to see some sort of viscosity reduction. And if you add in excess of your condensing agent, or perhaps specific types of condensing agents, then you can actually this can lead to precipitation, and you can crash out the DNA. So some of the structures that are seen, these condensed or collapsed structures, are rather than that sort of worm-like or random coil type state that I had previously discussed. One common one is that it would form this torus shape or toroidal shape, where now if you zoom in on this basically concentric rings that you have forming the torus, zoom in, now you see that you maintain the double helix, but now it sort of wraps around itself. And again, depending on the molecular weight of the DNA, this sort of torus structure can be composed of a single DNA or multiple DNA. Now, the driving force depends on the particular condensing agent that you use. So if you're using something to neutralize the charge, remember DNA is anionic or negatively charged, then you're going to reduce the electrostatic repulsion amongst the different portions of the chain, and then this can lead to the more compact structure. Also, if you change the solvent and reduce the quality of the solvent, so very similar thinking that you would have when you're dealing with synthetic polymers, in a good solvent, the polymer, or in this case the DNA, would be expanded, and as the solvent quality is reduced, and by solvent quality I just mean that the backbone segments, the relationship with the solvent becomes less favorable. So as you move towards something that's a poor solvent, then that molecule will collapse. So changing the solvent and not necessarily worrying too much about changing the charge can also be a driving force to create these condensed structures. So some of the common agents are multivalent cations of various sorts. So some common ones are a polyamine named spermine, which has a +4 charge. You can also have inorganic multivalent cations that introduce different collapsed structures. And then, so these ones are going to be more towards the charge neutralization to minimize electrostatics within the chain, so intramolecular statics or intra-electrostatics. Introducing alcohol into the water, then this is going to be more of a driving force going from a good solvent to a more poor solvent. And this then could be lead to these collapsed structures. So I've sort of highlighted this torus type shape, although there are different specific structures that can form, and the exact structures that will form will depend on the condensing agent that you use. I sort of highlighted this torus shape because it seems to be the common one formed when you introduce polyamines, and that's what we actually introduced in our experiments here. So, but bear in mind that there are some rod-like structures or more aggregate type structures that can exist as well. So then finally, the thermal transition. So this, in the end, we did a lot of our thermal transition experiments in the presence of some of these condensing agents, but you wouldn't have to. So what happens then when we start increasing the temperature of the DNA in solution? So we know that in aqueous solutions, as you increase temperature, this tends to disrupt hydrogen bonding. And so we talked a lot about earlier here that the nature of the DNA structure is about the hydrogen bonding, basically between two of the single strands to form the double-stranded helix. So the general thought, if you look in the literature as to what can happen as you increase the temperature, is that upon initial heating, you'll have some sort of partial separation of these double strands. And so not only will this lead to a more extended or expanded structure, but then it opens up the possibility that maybe these single strands on neighboring molecules can then meet up and have some weak hydrogen bonding, forming what we would think of as a physical crosslink. So sort of analogous to a gelation type behavior, if you're more familiar with that in the synthetic polymer world. Of course, then if you further heat your solution up, then this is going to just sort of at some point disrupt all hydrogen bonding together, and you move more towards single strand molecules in solution. So we might expect the viscosity then as we ramp up in temperature to be indicative of these transitions. And we'll share that data shortly. So in terms of reversibility, as I had mentioned, this isn't like the protein denaturation, which is when induced from a thermal stress is not reversible. This reformation of the double helix structure is theoretically fully reversible. However, it tends to be somewhat of a slow process to actually recreate these double-stranded helix structures. And it's going to depend on, how much it recovers will depend on the cooling history. Because it turns out that sort of optimal temperature or annealing temperature where these will reform most rapidly is not equal to the melting temperature. So you would have to spend, an adequate amount of time, in some cases hours or days, to actually fully, reverse your steps and get back to something similar to the initial state. So just bear that in mind when it comes to, looking at the heating versus cooling cycles. And so these transitions that occur, from the thermal, from the heating cooling cycles will be dependent on a variety of things, and this is part of what we'll discuss today, and that will be the DNA concentration, and then also what sort of buffer or environment is the DNA in. So is it something like DI water, or are you using a buffer with various levels of ionic strength and different types of counter ions? This is all going to impact, how this transition plays out. So with all that sort of introduction there, let me just give you a quick overview of what we'll discuss in the remaining time that we have here. So I do want to talk about our automated VROC Initium 1+, because as I mentioned, we're doing a lot of temperature sweeps, both heating and cooling up to 70 C. And I think I just wanted to highlight the nature of our system and how beneficial it is when you start doing these types of experiments, because we don't have to worry. The closed nature of the system means that we don't have to worry about evaporation. We don't have to take extra care to handle that. And so we're going to look at mainly the data we'll share is relative viscosity as a function of temperature during our heating and cooling cycles. And we're first going to look at the transitions of the DNA where we're just looking at how the concentration of the DNA itself influences this. How does this transition manifest differently depending on the shear rates that we're operating at, and how does the solvent composition impact these transitions? And then finally, we'll look at the impact of adding the condensing agent, the polyamine, multivalent polyamine, to see how this reduces the viscosity depending on the concentration of the DNA, and then also how does this condensing agent impact thermal transitions, basically the melting of the DNA structure. So just a quick overview then of our technology. So we have our VROC technology, which is viscometer, rheometer on a chip. So we combine microfluidics and MEMS. And so we basically have a flow channel where we control the volumetric flow rates in a well-defined flow channel, and we monitor the pressure drop across that flow channel with the sensors spaced along the base of that channel. So this is a controlled rate instrument where we're controlling the shear rates, we're monitoring the pressure, which allows us to calculate the shear stress, and we use both of these to calculate the steady shear viscosity. And just one final image here with our system. So this is a sort of a bird's eye view of our automated viscometer, the Initium. And I just wanted to emphasize that, once our sample is introduced into the test syringe, and that flow channel that I just mentioned is embedded into our VROC chip. Once our sample is introduced here, we have a closed system, and we have the ability to retrieve our sample. So when we do these heating cooling cycles, we have one volume of sample that we're monitoring in this closed environment over an extended period of time. So we are really looking at that same sample, and we know its history in terms of how it's been sheared and how it's been heated and cooled. And because it's closed, we know that it's not being, severely altered by any sort of evaporation. So we don't have to worry about any artifacts from that. Okay, with that said, I'm going to move on to Chrystian, who is going to talk about his experiments where we were looking at these thermal transitions and how the different concentrations of DNA and buffers impacted the data.
Thank you very much, Stacey. It's a pleasure to be here speaking in the forum today. So for the first set of experiments with the DNA solutions, we worked on solutions made with calf thymus DNA. So we dissolved this in different solvent compositions. So one of these solvents was just DI water. Another one contained Tris-HCl buffer, and the third one contained Hapes buffer. So Tris-HCl is a big component of PCR buffer, and another application is that it's used in gel electrophoresis. Now for Hapes buffer, this is widely used in cell cultures. So we prepare the solutions by combining these DNA fibers you see in the image here. We combine these with the solvent, and we let it rest for about 24 hours. For the higher concentrations, we actually let it rest for about at least 2 days. So for the samples with water, we prepared 7 DNA concentrations between 0.4 to 6 mg per ml. And we studied 2 shear rates and this temperature range of 40 to 70 degrees. Now for the samples with Tris or Hapes buffer, we focused only on 0.4 mg per ml and this higher shear rate of 22,000 inverse seconds. And we did this so that we could compare between the 3 solvent compositions at this matched DNA concentration and matched shear rates. So what exactly do we want to learn about these samples? We want to better understand how the viscosity is going to vary with the temperature for multiple DNA concentrations in DI water, and when you have other solvent compositions that are going to have different ionic concentrations or pH values, like Stacey mentioned. So what does this tell us about the structure of DNA in solution. That's something else we want to look at. So we performed temperature sweeps for DNA samples. And this plot here is showing you the relative viscosity versus temperature for one concentration of DNA, 4 mg per mil. So this relative viscosity is basically the viscosity divided by the viscosity of the solvent, so which is DI water in this case. So how we obtain this viscosity is that we cycle the temperature. So we first increase the temperature and then we decreased it. So you can see that by the arrows shown here. So basically we ramped the temperature up and then we ramped it down. So the top data that you see here is for the lower shear rate. And this bottom data is for the higher shear rate. So you can see that the viscosity is not fully recovered after one cycle of increasing, decreasing the temperature. And this is happening because the DNA is denaturing above a certain temperature. And here it corresponds to this viscosity rise that you see above around 50 degrees Celsius. So while this is happening, the DNA is melting and its double strands are pretty much dissociating and becoming single strands. So how do we quantify this melting? We do that by defining the melting temperature in this plot. So to define it, we take a sigmoidal fit of the temperature ramp up data only. So as only if the temperature is increasing. And we set the midpoint of this fit as the DNA melting temperature. And this is actually consistent with how it's defined in the literature. So the melting temperature is the same for both shear rates. So because of this, for the data that we're going to show you in the following slides, we're only going to focus on one shear rate. Another thing you can see is that it's easier to decipher, to identify this melting temperature for this lower shear rate data, this data on top. So as I mentioned before, the viscosity is not recovered as the temperature decreases back down. However, it's possible to anneal this DNA back into its double-stranded form and have you recover this viscosity by exposing your sample to its annealing temperature. And this would be done for some amount of time. For our DNA sample, that amount of time is probably in the order of days. And that's because it has a pretty high molecular weight, about 3 million. And we actually calculated this molecular weight using intrinsic viscosity analysis with our VROC instrument, and that app note was released, I think yesterday. So we extended the viscosity measurements to DNA concentrations between 0.4 and 6 mg per mil. So here we only show the results as we ramped up the temperature. So we basically omitted the temperature ramp down data. And this is so that the graph would not be so cluttered. So all this data for the lower is corresponding also to the lower shear rates, because as I mentioned before, this shear rate gives us a clear, more clearly shows us the viscosity rise and gives you a clearer melting transition. So in this graph, you can see that the viscosity rises greatly above 52 degrees Celsius. And as I mentioned before, above this temperature, the DNA starts to dissociate into the single strands. And another thing to see here is that the viscosity is rising more as the concentration of your sample increases. So if you look at the 0.4 and 1 mg per mil samples, it's hardly, this viscosity rise is hardly even noticeable. Actually, if you zoom in to these low concentrations, you will see this sigmoidal profile for these. So again, we take the sigmoidal fins and extract the melting temperature from the midpoint. And that's plotted here as a function of the concentration. So each error bar is 3 times the standard error given by the fins. And you can see that one of these error bars is much greater than the rest. So this is for the lowest concentration. And this is due to the larger noise for this much lower viscosity sample. And this will lead to analysis issues. So for concentrations 2 and higher, so in this graph you can see that the melting temperature is generally increasing with the concentration. And this agrees with many publications that they suggest that if you increase the DNA concentration, you're going to increase the melting temperature. So it's also known that increasing the salt concentration can increase your melting temperature. And here we're going to look at the relative viscosity versus temperature for 0.4 mg per mil DNA at this constant shear rate. So these 3 plots here in this one graph correspond to 3 different solvent compositions. So as I stated before, the solvents are 10 millimolar HAPEs in water, 10 millimolar Tris-HCl in water, and the third one is just pure water. So we can see that even though the viscosity is similar between water and the Tris buffer, the melting temperature is about 4 degrees larger for the Tris. So the melting temperature for the Tris sample is higher because you have this higher positive charge density that is neutralizing more of the negative charges on the DNA backbone. So this is going to lead to a more stable DNA molecule, and this molecule is going to withstand higher temperatures before it melts or before it denatures. So something interesting we saw is that the HAPEs buffer doesn't only just show you this much higher viscosity increase with temperature, but apparently this a lower melting temperature than even water. It's about 15-20 degrees slower than the other 2. So this HAPE solution, we know that it has a different pH and charge density than the other 2 solvents. And this may help, this is gonna help in explaining why it has a different melting temperature and higher viscosity. So this plot on the right is the same, except that we excluded the data for DNA and water, and that's for like more clarity, so that it wouldn't be so cluttered. We also added the data for the ramp down in temperature. So one difference is that the viscosity of HAPEs is not recovered after you ramping down the temperature. And this is consistent to what we saw for DNA and water in the previous slides. However, for Tris buffer, this viscosity is quickly recovered and matches for the ramp up and ramp down. So this is interesting because it's suggesting that the DNA in the HAPEs buffer is remaining mostly denatured as your temperature is ramped down, while the DNA in the Tris quickly anneals back to its original double-stranded form. So this indicates that the DNA is less likely to denature in Tris buffer. And this can have benefits in applications like PCR and gel electrophoresis. So we were also interested in how adding a condensing agent affects the viscosity and the melting temperature of our DNA samples. And my colleague here, Soha, is going to lead you through the results and analysis of that.
Thank you, Chrystian. So before I show you the data I've collected, I want to briefly explain the sample preparation, and after that I will show the viscosity data with you. In order to understand the effect of condensing agents, we prepared solutions containing DNA and condensing agent in DI water. The condensing agent we used for these experiments is called spermine. So we prepared 2 different DNA concentrations, including 2 and 6 mg per ml DNA, and then added various amounts of spermine to them. We measured the viscosity of DNA solutions containing different concentrations of spermine. And we studied one shear rate that is 2700 inverse second, and temperature range from 15 to 70 degrees C. What we want to learn from these experiments is to better understand how condensing agents can affect DNA structure in a temperature sweep experiment. And the good thing is that the changes in DNA structure in the presence of condensing agent can be monitored with viscosity measurements. And so our VROC initium is a reliable instrument for measuring viscosity that requires a small volume of DNA sample, which is important for us since DNA is relatively expensive. So after this quick review of sample preparation, I'm going to go over the viscosity data I collected from our VROC initium and share some results with you. This plot shows measured viscosities of aqueous 6 mg per ml DNA containing 100, 200, and 300 microgram per ml spermine over the extended temperature ranging from 15 to 70 degrees C in steps of 5 degrees C. It is evident from this plot that the addition of spermine decreased the viscosity. As seen, it is more pronounced after 50 degrees C. At this point, viscosity rises steeply to a maxima, followed by a gradual decrease. However, this gradual decrease in viscosity is seen for samples without spermine or a low concentration of spermine, I mean 100 microgram per ml. For the other 2 samples with higher concentration of spermine, this viscosity reduction shifted to higher temperature. Temperature higher than 70 degrees C, that is out of the initium range, and we are not able to monitor that. As my colleagues mentioned, the viscosity increase most likely occur because of DNA melting. Melting or denaturation temperature shifted to higher temperatures with increasing spermine concentration. As you know, in DNA structure, 2 single strands are bonded into 1 double helix, and at melting temperature, some of the hydrogen bonded double helix separate. So not the whole molecule separate. The hydrogen bonding disrupt partially and not entirely. So that many more crosslinks are formed than are possible with double helix DNA. And so the adjacent molecules start to cross-link together, and this increase in cross-linking, which is similar to the gelation of polymers, leads to an increase in viscosity. Now the question is that what happens after the viscosity maxima? The answer is that DNA melting and gelation dominate only in a limited temperature range, and at higher temperatures, the crosslinks break up due to the decreasing stability of hydrogen bonds. So as you see in this plot, viscosity drops at higher temperatures. With that in mind, now let's take a look at the relative viscosity versus temperature. The plot in the left shows the same samples as the previous slide and the corresponding sigmoidal fit for each curve. As Chrystian mentioned, we used sigmoidal fit to extract melting temperature. So the plot in the right shows melting temperature as a function of spermine concentration. I think now it's clear that with increasing concentration of spermine, the melting temperature shifts to higher temperature. For example, in the sample without spermine, the melting temperature is around 59 degrees C. However, in the 300 microgram per ml sample, melting temperature is around 66 degrees C. Spermine, as Stacey explained, is a cationic component when added to the DNA solution associated with negative charges on DNA structure and neutralize the charge. And so the internal electrostatic repulsion decreases and the molecule starts to collapse into a toroidal coiling structure, as Stacey mentioned. So DNA rapidly aggregates into the condensing or compact morphology, so the active volume fraction of DNA drops. So this is the mechanism why viscosity decreases in the presence of condensing agents. When the solution is heated up, for samples with higher concentration of spermine, more energy is required to first unfold or break up their toroidal structure. Or say overcome the condensing state and then overcome the hydrogen bonds in the double helix structure. So this is the reason why melting temperature increases with increasing condensing agent. We performed the same temperature sweep experiments with 2 mg per ml DNA and 5 different concentrations of spermine. The plot in the left shows the relative viscosity as a function of temperature. These samples show the same behavior or same trend as 6 mg per ml DNA, so that increasing the concentration of spermine increases the melting temperature and reduces the viscosity. Also, the plot in the right shows melting temperature as a function of concentration of spermine. As expected and as we discussed a few minutes ago, with increasing concentration of spermine, melting temperature increases. So just to quickly summarize, before we go over the questions, I just wanted to highlight that we showed how we used VROC Initium 1+ to characterize the viscosity and melting temperature of DNA solutions. It allows us to perform temperature sweep experiments with high accuracy and reliability using a small sample volume. We varied the DNA concentration, shear rate, the solvent composition, and the presence of the condenser spermine. And we showed that the melting temperature is constant between shear rates for the same sample. We also showed that increasing in the DNA concentration increases the melting temperature. And using the Tris-HCl buffer with higher ionic strength leads to a higher melting temperature but similar viscosity. Also, we showed that increasing the concentration of spermine increases the melting temperature and reduces the viscosity. With that, we will go over to the question section and see if you have any questions.
Anyone who has questions, please be sure to drop them in that chat panel, and our speakers will be happy to answer them. All right, RheoSense team. The first question is, does low versus high shear rates fundamentally and irreversibly change the DNA structure?
So the question being the impact of shear rate on, so I guess I might come back with the clarifying question. Is the question, can we denature the DNA by shearing it alone without any sort of thermal energy? I don't think so. If that is in fact what you're asking, we have not seen evidence of shear alone denaturing the DNA, at least not to rates that we can access. So unless I didn't quite get the question, that's.
That was the question, really. They clarified, yes, that was the question, and just wondering, you know, is it possible to run an increasing or decreasing shear rate ramp?
Yes, that's certainly possible. I don't think we, well, I guess we did do there were certain experiments where we did combine shear rate sweeps with temperature sweeps. So in some of the data that Chrystian was showing, he was just basically highlighting that point at where the molecule would unfold or lose its hydrogen bonding there was independent of shear rate, and he was focusing on sort of the extremes that we looked at. We could go to higher rates potentially if we looked at, you know, in our other instruments, our MVROC instruments, but up to the rates that we could access in the initium, we didn't see any denaturation alone from shearing alone. It had to be the temperature induced.
I'm going to give everyone another minute just in case there are any other questions. Another follow-up question, any correlation with thermogravimetric analysis, TGA, or similar math?
So we don't have internally, our only capability is the viscosity measurement. So we couldn't, we don't have any complementary data. However, I will say that, you know, the trends we saw were consistent with the literature values. We can't compare them exactly. We can't get a exact comparison because all of the behavior is going to depend on the molecular weight of the DNA. And some of the publications that we saw had, based on what the information they provided, seemed to be higher molecular weight DNA than what we had. So we had, I think, about 4,000 or so base pairs, whereas some of the data in the literature was maybe 10,000 plus. So we saw similar trends, but we couldn't get an exact comparison because of the sensitivity to the actual molecular weight of the DNA. But things that were consistent were things such as shift in melting temperature as a function of DNA concentration. So as you decrease DNA concentration, that melting temperature shifted to lower temperatures. And then also in comparison, when introducing the condensing agent, we saw similar behavior or similar trends in that the condensing agent reduced viscosity and also shifted the melting temperature.
Thank you for that answer, Stacey. And if anyone else has any questions, we still have a few more minutes. And if not, we will thank our wonderful speakers. Looks like at this time there are no more questions rolling in, so I would like to thank Stacey, Chrystian, and Soha for such a wonderful presentation. Thank you for being so thorough and for sharing your knowledge with us today.
All right, thanks everyone.
Thank you.
Thank you.